Hello and welcome. Today we're diving deep into the rulebook of our own biology, and we're going to see how nature just loves to break its own rules. We're talking about the hidden side of genetics.
Okay, so I want you to think back to high school biology for a second. Remember those Punnett squares? Yeah, those things. We all learned that when you do a dihybrid cross, you get this super neat and tidy 93 to 3 to 1 ratio. It was like perfect, simple, predictable. The only problem, it's not really the whole story. You see, in the real, messy, beautiful world of genetics, things are way more complex and honestly a lot more interesting. It turns out that simple ratio is more of a suggestion than a law, because nature is a constant rule breaker. So what's behind all this amazing complexity? Well, it's a concept called non-allelic gene interaction. This is when genes that are located at totally different spots on a chromosome start, well, talking to each other. They can work together, they can get in each other's way, and they can completely change the outcomes you'd expect. So let's jump right in with one of the most dramatic forms of this interaction, epistasis. You can think of it as the ultimate genetic plot twist, where one gene literally has the power to silence another one.
All right, here's the official breakdown. In epistasis, you've got a boss gene, that's the epistatic one, and it can completely mask or hide what another worker gene, the hypostatic one, is trying to do. It's basically a master override switch that can shut down other lights no matter what their individual switches say.
And this whole masking thing can play out in two main ways. With recessive epistasis, you need two copies of that recessive gene to pull off the mask. But for dominant epistasis, all it takes is one single copy of the dominant gene to do the job. And as you can see, this completely messes with the final ratios we end up seeing. So let's tackle dominant epistasis first. This is where a single dominant allele really does act like that all-powerful override, just completely shutting down whatever another gene is trying to express. Summer squash color is a perfect example of this. You've got one gene, we'll call it W. If a squash has the dominant W allele, it's white, period. But that W also masks any other color gene. It's only if you have the recessive WW pair that the second gene, Y, which is on a totally different part of the chromosome, even gets a say. Then, if that Y is dominant, you get yellow. If it's recessive, you get green. So what happens when you cross them? Well, you can forget that old 9-3-3-1 ratio. Instead, you get this wild 12 to 3 to 1 outcome. I mean, look at this. Any squash with just one of those dominant W genes, boom, it's white. That accounts for a whopping 12 out of the 16 possibilities. The only ones that even have a shot at being yellow or green are the ones that are totally missing that W gene. It just goes to show you the incredible power of a single epistatic gene. Okay, now let's flip the script and check out recessive epistasis. In this case, the mask isn't some powerful dominant gene. It's a sneaky pair of recessive ones. You need two copies like CC to silence the other gene. And this gives us another really unique ratio, 9 to 3 to 4. The classic example here is coat color in mice. You have a gene, C, that's kind of like a license for color. If a mouse has at least one dominant C, it's allowed to produce pigment. But if it has the two recessive CC genes, it's albino, pure white. That CC combination is epistatic to a whole other gene, the A gene, which decides if the color is agouti, which is a kind of banded pattern, or just solid black. So what's the final tally? This 9 to 3 to 4 ratio. You get 9 agouti mice and 3 black mice, which so far looks pretty standard. But here's the cool part. See that albino bar? That group of 4 is actually made up of mice that had the genetic code to be agouti and mice that had the code to be black. But their CC genes just masked it all, lumping them together into one all-white category.
Okay, but gene interaction isn't always about one gene bossing another one around. Sometimes they work together in really fascinating ways. It's not always a competition. Sometimes it's all about collaboration. A great example of this is something called duplicate genes. This is what happens when two totally separate genes can actually do the exact same job. As long as you have just one dominant allele for either of those genes, you get the trait. It's kind of like having a genetic backup system. Now, in the shepherd's purse plant, this leads to an absolutely wild 15 to 1 ratio. Two different genes can both create a triangular seed capsule. So the only way you can possibly get a different shape, oval, is if you have all recessive alleles for both genes. As you can imagine, this makes the triangular shape just totally dominant in the population, showing up 15 out of every 16 times.
And then you have polymorphic genes, where the teamwork gets even more creative. In this scenario, two genes can affect the same trait, sure, but when they're both present in their dominant forms, they team up to create a brand new, even better result. So in barley, a dominant A allele by itself will give you a medium length on, and a dominant B allele by itself will also give you a medium on. But when you have dominant A and B together, their effects combine to make a long on. And this gives us this very unique 9:6:1 ratio you see here. 9 long, 6 medium, and just 1 with no on at all. It's a perfect case of genetic synergy. So we've seen genes mask each other, and we've seen them collaborate. But the real genetic web is even more tangled than that. It goes beyond just how genes interact with each other, to how a single gene can have massive, widespread effects. And that brings us to a concept that just shatters that old one gene, one trait idea, pleiotropy. This is what we call it when a single gene creates a ripple effect, influencing multiple traits throughout the body that might not even seem related.
Just think about sickle cell anemia. One tiny mutation in a single gene changes the shape of red blood cells. That one change then causes a whole cascade of devastating problems. The same goes for PKU. One defective gene affects how the body processes an amino acid, which then leads to a huge range of severe symptoms. It's a really powerful reminder of why understanding these complex connections is so critical in medicine.
So if you take one thing away from all of this, let it be this. A single trait, whether it's the color of a flower or our own health, is almost never the result of a single gene, just doing its own thing in a vacuum.
So let's recap the hidden rules we've just uncovered, right? We've seen that genes can totally mask each other, like with epistasis and its funky ratios. We've seen they can have redundant roles, almost like a backup plan. And we've seen they can combine their powers to create brand new effects. The big takeaway here is that it's not some simple linear instruction manual. It's this incredibly complex network of teamwork and sometimes suppression. So instead of thinking about genetics as a simple blueprint, it's much more accurate to picture it as a massive ongoing conversation. A dynamic, interactive discussion happening between thousands and thousands of genes, all influencing each other to create the final incredible result. And I'll leave you with this final thought. Everything we talked about today, all of that amazing variety and complexity, came from the interaction of just two genes. So just take a second and imagine the truly mind-boggling complexity that comes from the conversation between the 20,000 plus genes that make you, you.